Good afternoon, everyone. Thank you for being here. As many others have said, it's fantastic to be back face-to-face and be among old friends and new friends. So what I'm here to tell you about today in the next few minutes is Catamaran Bio. We are a private Boston-based biotech working on engineering natural killer cells with the purpose of treating solid tumors, which we view as a very, very high unmet need. And over the next few minutes, I'll tell you about our approach there and why we think we'll be able to address these patients. So we've made significant advances, and over the course of this presentation, I'll tell you about our two pipeline programs. We are focused on treating HER2-expressing tumors, so those are gastric, breast, and ovarian cancers. And here we've established in vivo efficacy in an animal model, and we've also garnered positive feedback from the FDA. And so we have a high degree of confidence that we'll be able to get into the clinic and file the IND in the 2023 timeframe. We also have a program that's targeting Cd70. This is a target that is very high potential, expressed across a variety of tumors, and we've also been getting good in vivo data on our progress there. And beyond those first two targets, the platform can also be deployed against additional targets, and we think that over time, this will really turn into an IND engine. The platform we've dubbed Tailwind. It is an end-to-end CAR-NK design and manufacturing solution. We have been working on the manufacturing process that will allow us to ultimately deliver off-the-shelf cryopreserved product that can be administered at the clinic setting for patients. We're doing this through a non-viral engineering method that has significant advantages over the legacy systems, and we believe this will allow us to insert larger payloads and to be able to manufacture in a very efficient manner. We're also able to build in proprietary synthetic biology solutions that will allow us to tackle the challenges of the tumor microenvironment, which is key to addressing solid tumors. In terms of where we are as a company, we're about 45 people strong, heavily focused on R&D and manufacturing. We've raised a significant amount of capital to date, but are again in the capital raising mode, which, as you can imagine, in the current environment is just a lot of fun. So let me set up the problem for you. Fundamentally, we know that cell therapy has been transformative in the treatment of blood cancers like leukemia and lymphomas. But that success has not really translated to the solid tumor setting. And that's because of several problems. We know that the tumor microenvironment is very harsh for engineered cells. We know that tumor targeting is very important and that persistence of cells is an issue in the tumor microenvironment. And none of the existing cell therapies really address all of those concerns in concert. And that's where our Tailwind platform comes in. As I'll describe to you, we have some synthetic biology solutions that allow us to address the tumor microenvironment immunosuppression. We've designed CARs chimeric antigen receptors, that allow the NK cells to be able to find and traffic to the tumor. And then we built in cytokine support to enable those cells to then proliferate and have activity in the tumor. And all of these edits are made possible by our transposon technology. And some of the progress that we've made over the last 2 years of our company's existence, as I'll go through in a moment, we have demonstrated in vivo efficacy in a HER2 xenograft model. We've gotten favorable feedback, very constructive, from the FDA on how to advance this into first-in-patient studies. And we've ironed out a lot of the kinks in the manufacturing process. As you've heard from many folks, the process is the product, so we've spent a lot of time really making sure that at a research scale, we're able to manufacture these NK cells and deliver them in the way that we think will be effective for patients. So let me tell you briefly about the platform and the way we've constructed it. As I've mentioned before, we do not use a legacy viral system. Instead, we use the TC Buster transposon system. This is a technology that was in-licensed from a company called Bio-Techne, who's also represented here at the conference. We like it because it's very simple and cost-effective manufacturing, requires far fewer GMP components than a viral chain would necessitate. It also allows us to insert large genetic payloads. We know that for the multiple edits that we're looking to do for these NK cells, we need to have capacity in this vector to be able to make the multiple engineering. It also allows us to multiplex in the sense that we can simultaneously knock in and knock out genes in a single step. So we like the simplicity and the versatility of this platform. We've also demonstrated that we can use this with significant efficiency and effectiveness in NK cells. So in the graphic that you're seeing to the left, what we're demonstrating is that with multiple donors, we've been able to take the transposon and effectively engineer these cells with upwards of 50, 60, and even in some cases 70% plus efficiency. So this is so efficient, in fact, that we don't even need to run a selection step in our engineering. And not only are we able to engineer these NK cells, we've been able to show that these engineered NK cells are highly active. So in the graphic on the right, what you see is that the engineered cells, which are in the blue line at the top, have almost 100% cytotoxic activity as compared to the control NK cells that you see at the bottom. Our current process looks like this. I'm not going to go into a high level of detail, but what you can see is that we start with a single leukopak. So this is a peripheral blood-derived NK cell. We isolate them, expand them, and then ultimately have a cryopreserved product. And we believe that with our current technologies, we will be able to take a single manufacturing run, so a single leukopak as substrate, and be able to turn that into hundreds of doses that are available for clinical use. The product will ultimately look like this. We have built in a CAR, which is again the chimeric antigen receptor, that will allow targeting to our antigen of choice. We built in an IL-15 module for NK cell persistence. And we've also built in a tumor microenvironment solution in the form of a TGF-beta trap, which I'll tell you more about in a moment. And we believe that this constellation of edits is going to allow us to succeed in the solid tumor microenvironment. The pipeline, as I've mentioned already, we have two programs that I really consider to be co-lead programs, both moving towards the clinic in the 2023 and 2024 timeframe. The first is targeting HER2-expressing tumors, and the second is targeting CD70-expressing tumors. We chose these targets for a variety of reasons. I won't go into much detail on this, but we like HER2 in the sense that it's very well clinically validated. We know what the translational models need to look like, and there are sufficient animal models that are available to us to give us confidence in this approach. And we know that we are differentiated here because we are the only cell therapy solution that is addressing the solid tumor microenvironment by building in this TGF-beta trap. Similarly, for CD70, we have sorted out some of the technical issues that are involved in creating a CD70-directed cell therapy. We know, for instance, that CD70 is expressed on activated lymphocytes. So in order to actually make these cells, we have to both knock out CD70 and at the same time introduce a CD70 CAR. And we've been able to do that with our transposon platform. So just to give you a sense of some of the emerging data coming out of these two programs, CAT179, again, is targeting HER2. And on the right side, you see here in vitro cell killing in a SCOV3 model, which is a HER2-amplified ovarian cancer line. And you can see that the yellow line at bottom are untransduced NK cells. They have some natural killing ability, as the name would imply, and that's one of the reasons we like using NK cells as our cell chassis of choice. But you can see that once you engineer those cells, which is the blue line above, that you get radically enhanced killing of this cancer cell line. And so what that HER2 CAR is doing is conferring very robust and very target-specific cytotoxicity. This also translates well to the in vivo setting. So on this slide here, this is an experiment that our scientists have run using our CAT179 cells. We have run this in a SCOV3 xenograft model. So these tumors are inserted into immunocompromised mice, and we then treat those mice with our cells or with control cells. And what you can see is that there is a significant reduction in tumor burden with the CAT179 treated animals that you can see there on the blue line at the bottom. This is also reflected in the imaging that you see here. This is a luciferase-based model, so you can see the tumor lighting up in these animals. And in both the saline and the unmodified NK animals, you can see significant tumor burden. In the treated animals, you can see that there is almost no residual tumor burden. And that's also reflected in the survival of these animals, which I've not shown here, but essentially all of the treated animals survive and none of the untreated animals do. So this gives us a high degree of confidence in moving forward that with this HER2 program, we'll be able to treat breast and gastric cancers that express HER2. Very similarly, for our CD70 program, you can see here that we've engineered in a CD70 CAR, also the TGF-beta trap, and an IL-15 module. And in addition to that, we've also engineered in a Cd70 knockout so that in these cells, we can actually physically take out the CD70 that's expressed on the surface. And you can see that in vitro studies, which is in the graphic at the bottom right, you can see that, again, our CAT248 treated cells are able to demonstrate significant killing of a renal cell cancer line called 7860. So in the time that I have left, I'd also like to just give you a sense of some of the future directions. So we have also been thinking about additional ways to really be able to crack the tumor microenvironment. And our synthetic biology solutions here, I think, provide a really elegant solution to the tumor microenvironment. One of the areas that we're looking to impact is TGF-beta. So you may know that TGF-beta is very heavily expressed in solid tumors of epithelial origin. And it's one of the key ways that the tumors evade the immune system. The TGF-beta that the tumors secrete turn off the immune cells that are in the environment. And so you need to find a way to bypass that immunosuppressive signal. Our first generation switch is really a trap. So what this is a TGF-beta receptor on which the intracellular domain is truncated. And so it's able to take in the TGF-beta at the cell surface, but not transduce a signal. And so I'll show you data in a moment that shows that this makes these NK cells completely impervious to the effects of TGF-beta. Next generation, which I'll tell you more about in a moment, is also a way to actually think about making TGF-beta an activating signal for these NK cells. So turning the tumor's defenses into an offensive signal for the NK cell. The TGF-beta trap, as I mentioned a moment ago, is a way to mitigate the effect of TGF-beta. You see here on the left side in the gold bars, when you take NK cells and you administer TGF-beta to them, their ability to kill cells goes down by about 75%. When you do the same experiment with cells that we've engineered with our modified TGF-beta receptor, you can see that they have no effect whatsoever once they're treated with TGF-beta. So this has made them blind to the effect of TGF-beta, and they retain their killing activity. And we've moved this over to the in vivo setting on the right-hand side. This is a neuroblastoma model showing that when mice are treated with unmodified NKs, they have some benefit, but it's marginal. When they're treated with TGF-beta trap transduced NK cells, however, the tumor burden is significantly reduced and lifespan is extended. So all of this is exciting, but we think that it's even more exciting to think about what the next generation could include. And here we've taken our TGF-beta trap and we've done additional synthetic biology and additional computational biology approaches to really think about how we can transform that signal. And what we've done here on the left-hand side is by changing the intracellular domains, you can see that we can tweak the way TGF-beta actually impacts these cells. And in fact, we can turn TGF-beta into an expansion signal for these cells. And on the right-hand side, what this graph shows you is that we can do the same with various other tumor microenvironment factors. We know that the tumor microenvironment is highly complex. TGF-beta is not the only immunosuppressive signal, so it's going to be important for us to be able to show that we can have this effect with various other tumor microenvironment signals as well. So what this is giving us is a portfolio of very tunable receptors that will allow NK cells to not just survive, but thrive in the tumor microenvironment. So just to wrap up, we are advancing our lead programs towards IND-enabling studies. As I mentioned earlier, doing a Series B raise, and then in parallel with that, also exploring partnership opportunities. As you can imagine, the platform is versatile enough. There's more than we can prosecute on our own as a small company. So if there's any interest in discussing ways that we can work together, would be happy to connect with you. So thank you for your time.